before we go on to do pedigrees, let's review some material about Mendelian genetics that you should have gotten from Chapter 3 or some of the previous videos or the material we covered in class. And remember, Mendel's laws of inheritance are the result of meiosis. So when we're talking about meiosis, when you think about meiosis, you should be thinking about Mendel's laws and how they impact each other. For instance, in Mendel's laws of inheritance, the law of segregation is just the result of each chromosome being separated into each chromosome from each parent being separated into different gametes. So when you remember, we go from 2N to 1N. So as we have one mother and father chromosome, one chromosome we got from mom, one chromosome we got from dad, being separated into separate gametes. And this is that separation is called the law of segregation. This is what Mendel discovered when he looked at pea plants, but he didn't know they were chromosomes. He didn't know they were DNA. He called them factors. But the bottom line is that he realized that they were separated, that they separated into different gametes. We know that as meiosis. The law of independent assortment is just the fact that this separation is independent. It's independent of any other separation. And that law of independent assortment we know as, again, that separation that occurs in meiosis 1 of metaphase, of metaphase, where the chromosomes line up and they separate. And which chromosome ends up in which cell is random, and that's what we call independent assortment. They're not dependent on each other. The law of dominance talks about the idea that one gene can be dominant over another gene. And this is what Darwin worked with when he was looking at his data. It clearly showed that the purple color was dominant over the white, for example, or the tall was dominant over short. When he looked at the data that he collected, this is what allowed him to say these factors one factor can be dominant over the other. That's dominant and recessive. Those are the three laws that Mendel came up with, things that really haven't changed much. But today we know things are a little more complicated. And Mendel was very lucky that he looked at the traits he did, because there was no incomplete or codominance. And just to remind you what incomplete dominance is when you have a blending occurring. So incomplete is blending. And you can review the video that you were supposed to take notes on if you don't remember what blending was and what incomplete dominance was. We also have another relationship between genes, and that's codominance. Now, codominance is this idea that instead of blending, you get both. So when you're talking about codominance, you're talking about getting both characteristics, both phenotypes show up. So they both show up. So dominance is when one of the two alleles shows up, one or the other. And codominance is when they both show up, and blending is when you get something in between. So if it's dominant, if purple is dominant over white, then purple will be the only one showing up if both alleles are present. If you have codominance, then you get both showing up. You have purple spots on a white purple and white spots on the flower. So they would be both showing up. That would be codominant. Incomplete dominance is when you get a color in between purple and white, kind of a light purple in the flower. So those are the three ways that that genes can show their influences. When we're talking about pleiotropy, that's a whole nother concept, and that pleiotropy is when you see a gene that impacts more than one trait. So when you see pleiotropy happening, you see a gene that impacts more than one trait. For instance, the gene for PKU disease impacts more than one body system. The protein that it makes impacts more than one body system. When we're looking at an allelic series, that's when there's a series of alleles where one is more dominant than the other, but not dominant than a third. And there could be 4 or 5 genes, 4 or 5 alleles, and each one is relatively dominant over something else. heterozygous overdominance, and that overdominance just means that the heterozygous has an advantage over both the homozygous recessive and the homozygous dominant. It kind of is a little anti-intuitive, but the bottom line is that in overdominance you have a situation where the individual with a head that's heterozygous shows a stronger phenotype than even a double dominant homozygote. Again, an allelic series is when you have a series of genes, a series of alleles that where they have varying dominance over one another. Epistasis is when you have a gene that is where you have a gene that influences another gene. So one gene can in some way influence the gene product of another gene. And then you have polygenic traits, and those are, again, that shows you where one single gene can impact more than one. You have more. The a trait can be determined by more than one gene. So where epistasis is one gene impacts another gene, in polygenic traits you have more than one gene impacting a trait. And interestingly, pleiotropy is when you have one gene that impacts more than one trait, where polygenic is where a trait is determined by more than one gene. So these are all possible ways that genes interact with one another. It's important that you understand that when we talk about dominant and recessive and even X-linked, they're just the beginning. And then when we're talking about this series of different ways of complicated gene action, that they do exist, you need to know that they exist. The key types of these types of interaction are going to be incomplete dominance, codominance, obviously dominant and recessive, and possibly epistasis. I might see that show up. I would consider that a possibility showing up on your test. You might see any of these on your test in February, but I think these four are a good bet for possible test questions on your end of course exam. If I were going to make a bet, and if I were someone who wanted to limit them, I only had so much time, I would focus on these four, and then if I had a little extra time, I'd make sure I knew the rest. So genes and the environment, sex-influenced traits, environmental dependent gene expression, and incomplete penetrance are also things that happen in gene expression or gene action. So you can have genes that not only are they impacted by other alleles, depending on which allele you have. Not only may they depend on whether another gene in the DNA and even on another chromosome. Not only can one gene be codominant, not only can they be incompletely dominant, not only could also impact more than one trait, but on top of all that, the environment can also influence the genes themselves. So whether you're a male or female, different genes might be expressed in different ways or to different intensity. You might, in one environment, you might express one gene. In another environment, you might express another gene. And depending again on this homeostatic relationship that we have with our environment. In other words, is just like your thermostat. If it gets hot, your thermostat turns off the furnace. If it gets too cold, the thermostat turns on the furnace. So this relationship between the environment and the reaction of your body is like the thermostat that controls the heat in your home. If it just like your thermostat turns on the furnace when it gets too cold and turns it off when it gets too hot, your body also reacts to the environment by turning genes on and off. So just like in class, I want to make sure that you understand what the different symbols mean on a pedigree. And we just to make sure you realize and remember that this would be a square is a boy, a square that is filled in is a male that is diseased. A circle is a female. circle is a female. I'm trying to find a free circle here. So any circle is a female. A circle with a dot in it is a carrier, meaning remember, how do we carry things? How do you have a gene and not express it if one gene is dominant over the other? So if you were, in this case, you'd be a carrier of little a, where big A is is the gene that shows up. So you carry the little a, but you don't show it. So that's where we demonstrate that by drawing either a circle or a square, and we put a dot in the center. So the dot in the center is a carrier. In this case, you see that the only carriers are females. That's a clue. That's clue number one, is what's going on here. And in this case, you'll notice that only the males have the disease. So males have the disease more than females. So that's important. That's really going to be, and you'll see that that's an important trait when you're looking at pedigrees. When you're looking at these lines, if you follow the line down, everything that's connected by a dark straight line, they're definitely related in some way. So you see this male is the father of Albert, and this female, Victoria, married Albert. So then when you see a horizontal line connected male to female, that means a marriage. And this line coming down shows that they had children, and every square or circle connected to this line directly is a child of Albert and Victoria. So we come along, we see a female here, Victoria. Here, this male is connected to these two. There's a female. This here is an interesting notation. You'll see it has a kind of a loop. It's to demonstrate that this line is not directly connected to this line. So when you see this line here, this is a child of Victoria and Albert, but this one skipped over. This is because this male here was the child of this male, but not related to these two, you see. Why draw this line? Because it's trying to show you that this person really is in the third generation, should be here in the third generation, but skipped the generation to marry this lady here that you see here, Alice of Hesse. So this male is an older man that married a younger woman. Same thing here. This male also married a younger woman and married a woman of the fourth generation, even though he is a member of the third generation. And that goes on throughout. Question marks are whether we don't know if they had the disease or not. Perhaps they died early. Perhaps they moved away. There's no information on it. And that's it. That's pretty much how you read a pedigree. How you read it. Diseased. It means it's filled in. When it's not, it doesn't have the trait. If it's not filled in, it has the trait or disease. If it is filled in, and if it's a dot in the center, it's a carrier. So pedigrees are important because they help us. Analyzing pedigrees help us understand what is how does the disease get passed from one person to the next. As you saw, there are many different ways genes can interact with a body to show up. The phenotype can be inherited. The trait can be inherited in so many different ways. So when you're talking about pedigree analysis, it's much more useful than Punnett squares in many cases, because Punnett squares require a great deal of a very large sample size to be accurate, where pedigrees are just a record of what's happened, so they can actually lead us to a lot of information that perhaps we can't get from just a Punnett square. Punnett square is just letting us know what are the odds that any particular thing can happen, but it doesn't really give us any direct information. We can predict within some kind of quality. It gives us some kind of quantity as to what the odds are of something happening. But the problem is, with such a small sample size, it's possible that, for instance, to get all 5 of your children to have the particular trait, or it's possible that none of your children have all the traits, just in that particular small sample size. But if you look at the entire planet, then the Punnett square becomes much more useful. That's why Punnett squares are so useful in studying things like flies and fish that lay 10,000 eggs at a time or 1,000 eggs at a time, because that's 1,000 samples, where a human can usually only has 4 or 5 children, at least in our day and age, 4 or 5 children at a time, and that kind of limits our sample size. So doing a pedigree is really important for genetic counseling. It's really important to, as a useful tool predicting what your chances are of having a particular disease. So knowing what your uncles had, what your great uncles had, what your great grandmothers had, what runs in your family, what doesn't run in your family, recording that, getting that written down, and really analyzing that can be very useful in making health decisions in your own life. These are the basic patterns of inheritance that can be easily determined by pedigrees. There are others as well, but these are the 4 that are usually tested, and probably incredibly important that you get to know, and especially if you want to do well on your exams. And that's your autosomal recessive, your autosomal dominant patterns, X-linked recessive, and X-linked dominant. Even though X-linked dominant is very rare, if you'll notice the pattern, it's important that if you see the pattern, it's important to be able to make the decision on which of these 4 inheritance patterns you have when you're looking at a pedigree. Okay, so here you have cystic fibrosis in this particular one. Now, someone might say, well, wait a minute, there's no dots, there's only someone who has it and someone who doesn't, male or female. That's because that's the initial information any scientist is going to be able to gather from a parent. We're not, or from a patient. You're not going to be able to tell whether automatically, oh, she was a carrier, he wasn't a carrier. These things are not things that you can read. You have to determine it given a certain amount of facts. So, for instance, you're given that this particular male has it, and that this female has the disease. How do I know that they have the disease? Because they're filled in. So remember, if it's filled in, then that filled equals they have the trait. It's either the trait or disease, right? So I'm going to use trait because we don't have to use it for diseases. It could be just having the trait. But we'll go ahead and say, we'll say trait. But because this says cystic fibrosis, we're going to go ahead and say that we'll call it a disease in this example, but when we're looking at filled in or not filled in, it's not necessarily a disease. Please remember that. So if it's cystic fibrosis and you see that this person here has the disease, but neither parent has it, how is that possible? How can you possibly have an individual that has a disease, but how can this person inherit a gene that gives it a disease, but neither one of the parents have it? Well, if we stick to dominant recessive, then we know that these parents, that this disease has to be recessive. Because if it were dominant and this person had it, then one of the 2 parents would have to have the disease, right? So if it were big A and this person has a big A here, a big A or 2 big A's, then one of the 2 parents had to give it a big A. And if it got a big A from one of the parents, then one of the parents has to have the disease. But neither have the disease, so neither have that big A. So it couldn't give it to this kid. So what does that tell you? It's not dominant. Well, if it's not dominant, it has to be recessive. And if it's recessive, then we know that this is little a, which means that this is little a over here. All right. So for sure we have that genetic information. Now we've identified what alleles that these parents have. We know that we know the cystic fibrosis now. We know it's a recessive inheritance pattern. There's one boy that has it, one girl, so equal number of boys and girls, so they're not sex linked, right? So sex is not important for the inheritance of the disease or trait, so it's not sex linked. So if it's not sex linked, then we would call that autosomal. Autosomal means of the not sex cells or not sex chromosomes, so they're autosomal. So these are passed on by on a gene. The gene is on one of the 22 chromosomes that are not the X or the Y. Okay, so we know it's recessive, and we know it's not sex-linked. We know it's recessive autosomal. That's the inheritance pattern. We know that this particular individual's little a. We know this individual's little a. So what do we know anything else? Well, we know that this parent has to have a little a because it had to give a little a to its child, and we know that this parent also has to have a little a because it gave a little a to its child as well. So both these two little a's here had to come, one from one parent, one from the other, so they had to have little a. We know that they have big A because they don't have the disease. So they have big A, but they don't. And they don't have the disease. So because they don't have the disease, they have a big A. So we know that they have big A, little a. That we know they're heterozygotes. This female here, and this female here, and this female here, they have to have a big A because they don't have the disease, and they have to have this lady has to have a big A because she doesn't have a disease. We're not quite sure yet what the other allele is. But as we look at these other individuals, look at this child, and down here, this generation, we notice there's a distinct difference between this group, and that is that this group has an individual that's diseased. So what that automatically tells you, this child having the disease tells you this is little a, but it also tells you that this parent had a little a and that this parent had a little a. So when we look at the inheritance pattern, what possible genes this person could have, we know that this person has to have a big A as well, because it doesn't have the disease, it has to have a big A. We know it also has a little a because one of its children has a disease. All three of these all have big A's. We're not sure what the others could be. And over here on this side, we know that all three of these have a big A, and we don't know what the other gene is. We know this person also has a big A. Now, if we did a Punnett square really quickly, and we said big A, little a, and big A, little a, again, looking at probabilities, just looking at the probabilities of what we would get if we cross these two. What is a random probability tell us? That we get 1/4 of these would be little a, little a. Indeed, we got that. And we would see that half of them would be big A, little a, and then 1/4 would be big A, big A. So we would guess that, and it would have to be random, that one of the three of these, so the guess would be that one of the three of these would be homozygous dominant. So we'd have one big A, and there'd be two of them would be big A, little a. That's what we would guess. And over here, when we're looking at this, none of them have the disease. There's three babies. Then we would guess that this person here has big A. That would maximize your chances, right, of getting no babies with the disease. And then this person has a 50-50 chance, right, has about 50% chance. There's a 50% chance that it is, well, it's actually, I should say not 50%. What I should say is that this person has a 2/3 of a chance of being big A, because we know it's not little a, little a. We know that it's not that. So out of the last three, there's a 2/3 chance it's a big A, little a. So it's probably big A, little a. And then what are the odds of getting, if we do that cross here on this Punnett square, what are the odds of these parents producing producing babies without the disease, you see that we have a 100% chance of having babies without the disease, but there's a 50-50 chance of whether these three are big A, little a, or big A, big A. It's 50-50. So 50% chance each. So that's a whole analysis of this pedigree on what we might get. So if you were this person here, you're going to a doctor's office, you're this person here, you want to know what are your odds without doing any DNA testing, what are the odds that your child would get this disease. It would depend on whether you were the one that was big A, or you're one of the two that's big A, little a. All right, let's go on and take a look at the next section. So that was autosomal recessive traits, and this was the what the chart looked like. And it's the key to autosomal recessive traits is they're rare, and a trait is rare. When you look at a pedigree, it's not going to happen often, or it's not going to happen as often as autosomal dominant. The trait skips generations, so you see here this generation does not have it, while this generation does. This generation does happen to have it, but you notice it also skipped this side of the family. So you get skipping in generations where autosomal dominant would not skip generations. The trait affects males and females equally. You see it has the circles and the squares. There's 50-50 on the pedigree. There's half the people have it, half the people that have it are male, half the people that have it are female. So that would be autosomal. That part is what makes it autosomal, and recessive is the skipping of the generations. So this is what determines it's recessive, where this part makes it autosomal. This also makes it recessive, that it's rare. So these are some common autosomal recessive diseases, and it's important to remember that autosomal recessive diseases generally are rare. And the question is, why are these autosomal recessive diseases in humans so common? And it's because they end up having the heterozygote gives the individual that is a heterozygote has an advantage. And it has an advantage for survival. Specifically, this advantage comes in with malaria. So malaria is a parasitic disease. It can be found in any swampy areas in the Mediterranean. It's found in Africa. It's found in the New World. So this malaria is a real problem for the world, has been for the human population in the world for a very long time. And it's not just humans; it impacts our food sources as it attacks cows and goats. So the advantage of having an individual that is heterozygote is that they're able to resist malaria better. So the heterozygote has an advantage over the homozygote dominant or the homozygote recessive. The homozygote recessive, for instance, in the case of sickle cell anemia, the homozygote recessive, that individual has sickle cell disease. The homozygous dominant doesn't get the sickle cell disease, but they do become sick from malaria. They're susceptible to malaria. So it's interesting that this is a prime example of what we were talking about earlier about overdominance. In that the overdominant, the heterozygote has an advantage over the homozygote, either recessive or dominant. So in this case, these diseases are good examples of this idea of overdominance, this idea that the heterozygote has an advantage over the other two possible outcomes. So when we're looking at autosomal dominant pedigrees, you'll notice that, and this is an example of it, what are some things that you might notice here that were different from the recessive pedigree? Well, I hope you said that on this pedigree, you're not skipping a generation. Every generation has the disease. You'll also notice that that is a consequence of everyone that has a disease has a parent that has a disease. You never get the disease without having a parent that has it. You can get people that don't have it. For instance, this child did not get the disease, but that's because they didn't get the big A. So in this particular example, like we'll go ahead and we'll keep talking about big A and little a. But in this particular example, let's say that that this is autosomal dominant, as we know already because it says it right here. But then that means that this individual here has a big A. It could have and how do I know that this individual then also has a little a? How do I know that this person is big A, little a? How do I know that? Well, I hope you said you know that. I know that because as I look at their children, the marriage between these two, it turns out that both of their children have it. Where marriage between these two, she has some children that don't have it. 3 out of her 5, 1, 2, 3, 4, 5 children, out of her 5 children, only 2 of them have it. 3 don't. So she must have a little a because 3 of her children must be little a, little a. How do I know they're little a, little a? Because they don't have the disease. If they had the disease, they'd have to have a big A. So it's key that you understand the difference between dominant and recessive. No, man. So the idea here is that when you're looking at this child here and this child here and this child here, they all have little a because they don't have the disease. Why? If they had to, in order to have the disease, you have to have the big A. That's what dominant means, right? So that also means that when you look at this person here, this person has to have a big A, and this person has to have a big A. But when we look at their children, they do have children. This female has children that don't have the disease. So they have to have a small a as well. These 3 children don't have the disease at all. So the more likely, and of course we know that the father must be little a, and this father must be little a, little a. They have to be because they don't have the disease. And here this father must be little a, little a. This mother here doesn't have the disease, but this one here doesn't have. This mother here has children that don't have the disease, some that do. So again, she has to have a little a to give, so she's big A, little a. And all her children, I'm not going to write it in because it's so small, but all these children here must be all little a, and this person here must have a big A, and in fact has a big A, little a. How do I know that this person has a big A, little a? Because dad only had a little a to give. Mom gave that girl a big A, but dad could only give a little a. In this case here, this again is true. Dad is little a, so mom gave this girl a big A, dad could only give a little a. This female here is big A, little a, again for the same reason, not just the fact that she had children that were little a, but also the fact that her dad was little a. So we can do that for the whole chart. We could fill up all the genotypes for this gene because autosomal dominant leads itself to know that if you don't have the disease, you're homozygous recessive, and remember that means little a, and that's when you don't have the disease. And if you have a father that didn't have it, or you have a child that doesn't have it, a father or mother that doesn't have it, or you have a child that doesn't have it, then you must be heterozygote, right? Because if you have it, then and your husband has it, you're homozygous dominant, and you have it, then all your children will have it because you only can give a big A. So no one, the only people that might be, in fact, the only person that might be homozygous dominant, the only one that might be, would have been, maybe had been homozygous dominant, would have been this woman here, but she did have some children who didn't have it on this side when she remarried. She married here, both her children had a disease. Then she remarried over here, and only these two had the disease. So that says that she does have the gene for the non-disease, as is evident from this male child, and this female child. All right, so that's how you would analyze a pedigree chart for an autosomal dominant. It's very the disease is very common. That's key to this type of inheritance. The disease is very common. It's found in every generation, doesn't skip a generation, and the affected individuals, they give it the autosomal part. That's this part here. The autosomal part means that you have equal male and females getting it. And if you look at the distribution of it, just about as many males have it as females. So there's no sex advantage. So it's not sex linked. Autosomal dominant traits. There are a few autosomal dominant human diseases, but some rare traits have this inheritance pattern. Skeletal disorders causing dwarfism is one example. One form of dwarfism is autosomal dominant. And why would autosomal dominant diseases, why would they be rare? That's a good question. And I didn't ask it in class because we didn't really have a lot of time in a couple of two of the classes. In the third class, I think I'll ask it today. So why is it that autosomal dominant diseases are so rare in humans? Well, mainly because if it's autosomal dominant, that means you're gonna get the disease if you have the gene, and if the disease is bad enough, it either kills you or makes you it somehow impacts your ability to reproduce. That gene is not going to be carried to the next generation. So a genetically inherited autosomal dominant disease is probably not going to continue from generation to generation, especially if it impacts the ability for the individual to have babies, to pass it on to the next generation. Now, X-linked recessive pedigrees are interesting. What do you notice here in this pattern that is different from the autosomal recessive? Well, first, what's similar? I hope you said they skip generations. So just by skipping generations, you know it's recessive. So we've identified it's recessive as soon as you see the fact that we have skipped a generation. Now, when you what makes it X-linked? What makes you think, or what gives you the clue that it's X-linked? It's found on the X chromosome. Well, I hope you say that only males are getting the disease. Since only males are getting the disease, one would think that there has to be some kind of sex-linked inheritance, and in fact there is. It means it's X-linked. The trait is rare, and that rareness comes from the fact that it's recessive. The trait skip generations, and that also gives you the clue that it's recessive. The fathers do not pass it on to their sons. That's a real key to X-linked. If you see a father giving it to a son, then it's Y-linked. And if you see that, and only if males get it more often than females, well, first off, it's Y-linked, only males are going to have it. But also the fact that this person has it, and he has a son that doesn't have it, you know it's X-linked and not Y-linked, right? Because this number one gives all his sons his Y, so all these sons have the Y. So if he has a disease, why don't these have? And why don't these have? These two sons have the disease. These are not his sons. They married into the family. So these two sons don't have the disease. That means it's not Y-linked. And the fact that only boys get it, only boys on this pedigree have it, makes you think that it's X-linked. All right. Anytime you have more males having it than females, you know it's X-linked, or you suspect it's X-linked. Anyways. All right. So we're dealing with X-linked recessive traits here, and this is the pedigree we began with the lesson with. We're talking about hemophilia. In the European royal family, hemophilia is something that runs through the family. We have carriers, and here's the dot that shows you a carrier. And here's this person doesn't have the disease at all. This person has the disease. And someone noticed, well, none of the people that had the disease had children. That might be because they died before they were able to have children, for whatever reason. In any case, when you look at this, and I hope you can see that more males than females have it. And in fact, in this pedigree, only males have it. The females carry it, and there's no male that gives his child the disease. So it's interesting. So it's not Y-linked. Females carry it, so it has to be X-linked, and more males than females have the disease. All right. So for sure, what we're dealing with is a series of information that we've gathered, historical information we've gathered and put in this chart. And someone asked me what the 3s and the 2s meant. They're right here in this legend. The 3, it means there's 3 females. 2 means there's 2 males. 2 females here, 3 males here, etc. And I'm not sure what the question mark means. It might be they don't know if the person had it or not, I suppose. Oh, here, uncertain status is what it means. They're not sure if the person had hemophilia or not. So there's some historical ambiguity to these people here. All right, so when we're dealing with X-linked recessive inheritance, dominant inheritance, we hopefully see that this is something that makes sense and you're able to do when you get your practice problems in class and on the exam. So this is a glucose 6-phosphate dehydrogenase deficiency causes the hemolytic disorder. It causes a hemolytic disorder. It's often fatal. It makes you sensitive to fava beans. It's the most common enzyme disorder worldwide, especially in those in Mediterranean ancestry. And again, the heterozygote it might give you an advantage over malaria. So having this disorder might give you not having the disorder, but having the heterozygous genotype for this disorder might give you resistance to malaria. Even though most people that get the disease die, right? As you see, often fatal, or not most, but often this is fatal, so that's not an advantage. But having the heterozygote, having the heterozygous or having the trait but not the disease gives you an advantage. And you might say, well, that doesn't make sense. Well, why would that? How could that possibly happen? If you think about it, if you look at the Punnett square again, looking at the Punnett square of it, the chances. When we look at chances, if you have the trait, if you and your wife or husband have the trait, then if you don't have the disease, versus having the trait versus having the disease, X-linked recessive. So it's going to be a boy. This would be these genes are on the X, right? Either you have it. Well, you know what? Since it's X-linked, why don't we go ahead and it's X-linked. Let's go ahead and do the X-link. So if you do a Punnett square and you do dad and mom, so having the trait, I guess you have the trait, you have the disease. So there's none of that heterozygous business here. You can't be heterozygote. So if mom, that's not going to work because you got 2 males having a baby. That just doesn't happen, right? So XX. So one of mom's genes has the disease. Let's just put an A. That's recessive. So let's say little a, and one is big A. It's not. And So one, if dad doesn't have it, you do have this heterozygote because the female is heterozygote. If dad doesn't have it and mom is heterozygote, then you have this going on. And what happens is that these two individuals here are susceptible to malaria, all right, where these two individuals here are going to be, if they survive, are going to be resistant. And in places where historically malaria has been a problem, this is a big advantage. So it's a big enough advantage that the disease is still around. And it's still one of the most, in fact, is the most common enzyme disorder worldwide. And that, again, is because this gives it either the heterozygote in the case of a female, or the individual that has the disorder in the case of a male. Even those, of course, only those that survive, because it's often fatal in males. Having the heterozygote gives you an advantage, so that the gene stays in the population because there's an advantage to having it, and those people survive and have babies, and the gene continues. So when you look at this dehydrogenase spread, you see it's all over the world. It's interesting that it's not just in the Mediterranean here, it's also in Africa. It's all throughout Southern Asia. It's all anywhere that it's wet and hot, because wherever it's wet and hot, you're going to have mosquitoes, and where you have mosquitoes, you have malaria, and therefore the gene is present. X-linked dominant traits. What you noticed is that it's common in the pedigree. When you look at the pedigree, that's going to be everywhere. Fathers pass it on to all their daughters. So you notice that when it's X-linked dominant, if you have a father, every one of his daughters have it. And males and females are equally likely to be affected when it's X-dominant. X-linked dominant is when both males and females are equally likely to get it, because, again, if it's dominant now, you don't have the advantage of that second X. So in the case of X-linked recessive, if you were female, you had the option, you had the second X that might give you, might let you get the dominant version, which is not diseased. But in this case, when the dominant version is diseased, you don't have that second X to back you up to help you resist that disease. So the fact that the X-linked dominant, all the females have it because it's dominant. All you need is one big A to get the disease, and of course, the female will pass. The female has a chance to pass it on to the son as well, and the female can pass it on to the daughter because it's dominant. Even though dad will give him his non-disease X, give her the non-disease X, she's also going to get mom's diseased X. So let's look at the Punnett square to explain this, okay? So to explain this one here, let's look at it and say you have an X with dominant A, big A. That means it's diseased in this case. Remember, every case, every Punnett square is different, depends on the situation. You have an X that has a big A. She has, actually, let's just do it traditionally and go with the dad on top. So, number one, X, no disease, little a, with a Y, that's dad. And over here you have mom, which is X big A, because she's diseased. She has the X big A. Even if she's heterozygote, which she is, because dad over here, her dad was not diseased. So this is a little a. This female has it. Why does this female have it? Because she has a big A from mom. Even though she has a little a from dad, that doesn't help her because it's dominant. If this were X-linked recessive, that extra X would have helped her. And of course, having this son also has it, right? Because he has a big egg that he got from mom. Even though he had a Y from dad, the Y doesn't help. It doesn't help at all because you have the dominant gene. Here, again, you have the recessive. So this person is not diseased because the disease is X-linked dominant, and this son is not diseased. So what you get is 50-50, half the boys, half the girls. So look at the boys. Half the boys have it, the other half don't. Again, half the boys have it, the other half don't, right? And half the girls have it, and the other half don't. So it's 50-50 when you're talking about X-linked dominant, so it's a little different from X-linked recessive. X-linked dominant traits are extremely unusual. Often they're lethal. They're before birth, so they're not even found. So when you look at X-linked traits in humans, they're pretty much you're pretty much dealing with X-linked recessive genes. But there are examples where some people are born and they're able to they have X-linked dominant genes. X-linked dominant diseases. So you have these two particular diseases are examples of people being born with them, and they're pretty serious. They're extremely unusual. They're rare. They're difficult to survive. But there you have it. All right, so let's take a look at an incomplete dominance. Although it's a real-life complication. Okay, when we're looking at real-life pedigree analysis, it really gets complicated because, someone with breast cancer, the breast cancer gene BRCA1 or BRCA2, has a higher probability of getting breast cancer, but it's not guaranteed. So they might have the gene BRCA1 and BRCA2, those particular alleles, but that just means they have a tendency to get cancer, not that they will get cancer. So we call this incomplete penetrance, and it probably has something to do with that there's an environmental impact, or that there's some other gene impacting whether you get whether you actually get cancer if you have BRCA1 or BRCA2. We don't know, but the bottom line is that it's hard to do a pedigree analysis for someone for these particular genes. Then you have the issue of sex-limited expression of genes that are only found in males or only found in females. Even though the female might have the gene only shows up in males. This is an issue when you're doing a pedigree, so something you would need to think about. Now, these are exceptions. You're probably not going to see them on your standardized exams, but it's good to know in the background. It answers a lot of your questions. Well, what if? Well, when the what ifs come up, and they do exist, and here's some examples, we have to find alternative ways of dealing with the problem. So remember, dominant traits may be rare in a population, depending on if they're diseases. Dominant diseases tend to be rare in a population. Recessive traits can be common, or they can be recessive, even though most of the time, if it's a recessive disease, it's going to be not so common, unless they give you some kind of advantage, right? Like the ones we've talked about already throughout the video. And often traits are more complex, and they involve the environment, or they involve other genes. So things are not always as simple as the problems we give you in class or we give you on a standardized exam, but it's good to be able to answer these questions. It's important to understand that. All right, so what is the pattern of inheritance that we're looking at here? Well, I hope you notice that it has skipped generations, so we're probably looking at something that's recessive, right? You got equal boy and girl getting the information, or getting the disease. So here we're going to call it autosomal. So autosomal recessive disease. Now, the question is, what are what is 4-2's odds of being a carrier? What's autosomal recessive, which means that this person is little a, this person is little a. This person must have a little a, this person must have a little a, they must have a big A and a big A. And if we break it down, big A, little a, big A, little a, and we do the Punnett square quickly, we see, and you can see you can do it pretty quickly. And what are the chances of being a carrier, being heterozygote? It's about 50%. So it's a 50% chance that this person is a carrier. And remember, carrier means heterozygote. All right, let's go ahead and look at the next problem. Sample pedigree cystic fibrosis. That's the cystic fibrosis one we did earlier. So what can you say about 1-1? Well, I think I already did these with you in class, so I'm going to let you go ahead. I did it with you in the video earlier, so I'm going to let you go ahead and answer these. I'll give you a few minutes a minute or so on the video, and then you can check your answers by rewinding the video as far as what we can say about each of these. You can say a lot about every one of these. All right. Just to give you a clue before the minute's over, first determine, is it dominant or recessive? And then, is it autosomal or is it sex linked? And then go ahead and fill in what you know based on what we just said. So basically that's just doing 3 steps. 1, 2, 3. One, two, three. All right, let's go on. What is this inheritance pattern? Well, you skipped a generation here, and you skipped a generation here. This one person has it, these two don't. This person has it, these two don't. This person has it, these two don't. So that tells you automatically it's recessive. Only boys have it. Boy, boy, boy. No girls have it. So it's X-linked. X-linked recessive. So what is the genotype of 3-1? 3-1 was the genotype of this person here, third generation, first person in that generation. What is this person? It's recessive, so you know that, and it's a boy. He got the Y from dad, so for sure he has an X. He got Y from dad, so so dad gave him a Y. His X is for sure a big A, right? Because mom has a big A. She probably has 2 big A's. Why do I say she probably has 2 big A's? She definitely has 1. Why do I say she probably has 2? Because none of her progeny have it, right? What about 3-2? Now this female definitely has a little a, and she definitely has a big A. How do I know she has a big A? She doesn't have the disease. I don't know if she has a little a on her X, because dad only has an X disease, little a, Y. So he could only, if he gives an X, she's a female, because he determines the sex. The Y, the little a comes down, so she definitely is a carrier. Also, if you look at the other piece of evidence, is this person here has it, so that means he got her X, his X from his mom, so that definitely means that his X little a came from his mom, whereas Y came from dad, right? And obviously this young lady here definitely got a normal X because she got her normal X from dad, but then her second X is a 50-50 chance whether she got a big A or a little a. We don't know. So what is it? And for 3, so 2 second level, I'm sorry, let's see, 2, 3, 2, 3, 2, 1, 2, I suppose that means this one. I don't see a 2, 3. But if they mean this one, then it's obvious. I think it's obvious it's X. Well, you can do this with all of them. I'm not going to go through all of them, but it's big A. How do I know it's not a recessive? Because he'd have the disease if it was recessive. And you can do that with each and every one of these. Either come up with an actual result or come up with a question, and do some probabilities if you want to with each of them. So this is an interesting question. We're trying to breed a black lab, and we're going to try to breed a black lab that's true breeding, which means every generation after that is going to get the same characteristics. And the two characteristics we're looking for are fetching well and not drooling. If they're gray, they're not drooling. If they fetch well, they're black. So, for instance, this person here this female fetches well and doesn't drool. There's 3 of them. There's 2 in this generation that fetch well, but they drool. See, there's no gray. They mated this male dog was mated with this female. This male is this female's uncle, but that doesn't matter because they're dogs, and so we can crossbreed them. So this male dog here was mated with this female, and it doesn't drool and it fetches, and this one does drool but fetches well. And our question is, how do we get true breeding line for both fetching well and not drooling? Well, these are the ones we have right now. I'm not sure if in this problem we can come and access the previous generations as well, but these are the ones we have now. This fetches well and drools. This fetches well and drools. This one fetches well and drools. So first, let's determine: is fetching well, is it recessive or dominant? Well, first of all, I see it everywhere, but it skips a generation here. You see it here, but you don't see it down here. Here you see it in both. You see it in all their children, and they're seeing all their children. So once that both parents have it, everyone in that lineage has it, as far as fetching well. And here you see it skip a generation. So fetching well is recessive. So fetching well is recessive. And males and females both do it well, so it's autosomal. Autosomal recessive. Gray individuals don't drool. All right, so when we're looking at gray, is that recessive or dominant? Well, here you have it, but here you don't. Here you have it. Here you have it. Here you have it. Here you have it their offspring, but this offspring has 2 that don't have it, but they do show up in the population. Hard to tell. I don't see it skipping a generation. It's common, but I don't really see it skipping any generation. So I'm going to go ahead and say that gray individuals also are gray. Looks like it's actually a dominant trait. Let's see if it was dominant here. It looks like it's autosomal. Looks like it's autosomal dominant because both males and females both have it. So I would say it's dominant and autosomal. So here we have it. On this side, it looks like we have they're all recessive. So these all have the recessive trait. This has the dominant trait. The key is to try to get a purebred dominant. So what we would do is we would cross probably this male with this female, and that would definitely give you black individuals. They would fetch well, but the question is this female heterozygote or homozygote? and obviously this female's heterozygote. So this may not be the best choice for choosing to mate with one of the males in this generation. This male here is also heterozygote, however, and doesn't look like we have any homozygote at all for this trait. Because why do I say this is heterozygote? Why do I say all these are heterozygote? If they're dominant autosomal, then that means that this parent here is recessive, which means that all his children have at least one little a, right? And all his children have at least one little a, right? All her children have at least one little a. So they're all heterozygote. So the key would be to keep breeding. How would we do it? We would keep breeding the black gray with each other until we only get black grays. We don't get any whites, whites rather than gray. All right. So we only get black grays. We don't get any black whites. And if we can do that, as long as we don't get black whites and we're getting all black grays, we keep interbreeding. So keep breeding this one, until all we get is black gray. Then we're sure we're getting the homozygous big A for the autosomal dominant trait of gray individuals, but you also are going to get big B, little b, because this is a recessive trait. Big B is incorrect. Let's see if I can get rid of this. And make sure that the dominant, that we cross them with each other, and that we know that the recessive trait of fetching well. Let's change it to fetching well. We'll say it's recessive, so it's little f. It's fetching well. And gray individuals, let's say big G is dominant, so big G, right? For drooling, so they don't drool, they get big G. We have to keep interbreeding them, keep breeding uncle with this cousin with this cousin until we get a series of puppies that only don't drool and fetch well. And in that case, we would have purebreds of homozygous recessive fetching well and homozygous dominant not drooling. Okay, that's it for pedigrees. You'll have some practice problems in class. You need to be able to analyze these pedigree charts as best you can. And please make sure you know your different ways of inheriting a gene and how they interact with one another and with the traits that they're responsible for. All right, have a good one.